Hi, good morning, everyone. Welcome to our last lecture. Let me know if you're seeing this, the broadcast. And also let me know if you were able to take a look at the video that I posted last evening. It was late, but it was on epigenetics. So let me know if you were able to view that. And if you did, here's the first question. What is epigenetics? What is epigenetics? Clementine, are you here? I sent you the links via your email. So let me know. Let's see. Yeah. Let me know if you're here. Okay. Okay, great. Okay, so I need an answer to my question. What is epigenetics? Did anyone look at the video? Okay, I will, let's see. Okay. Okay, well, it could be because I posted it late, so I apologize for the late post, but you could look at it after the lecture. So I'll please view after lecture. Tamatop, the video is on the Facebook page. It's entitled Epigenetics in Nova Science. Okay then, well, let's get started. I am going to share my screen. Oh, welcome Clementine, I see you now. Okay, I will share my screen. Let's see here. So we'll pick up from where we ended on Monday. Yeah. Okay, let's see. Rayford answered the question you said. Epigenetics is gene modification that allows or hides genes from being expressed via methylation or acetylation. Well, yes, those are methylation and acetylation are some of the techniques or mechanisms of epigenetics. But epigenetic, what is it? It is changes in gene expression that is not dictated by the gene sequence, but by the methylation and acetylation that can occur on the DNA. Okay? So DNA changes that are not governed, or DNA gene expression that is not governed by the DNA sequence. Okay, okay, let's move on to, let's start the lecture. And I'm going to share my screen. Let's see here. There we go. Start screen share. Okay, so on Monday we started looking at regulation of gene expression in eukaryotes, and we saw that it's much more complex than prokaryotes, and that there are cis-acting and trans-acting elements involved. The cis-acting elements are the promoter and the enhancer. The promoter is bound by RNA polymerase, and in this chapter we're focusing on RNA polymerase 2, which binds the promoter. The enhancer, on the other hand, is bound by transcription factors, and there are different types of transcription factors. We have basal transcription factors, which allows for basal level gene expression or low level gene expression. You then have activators, which increase gene expression above the basal limit, and then you have repressors, which would lower gene expression. Repressors can function in different ways to modulate gene expression. They can function by competition with the activator. They can function by quenching the activator, that is blocking the activation domain of the activator. They could also function by sequestration, cytosolic sequestration, preventing the activator from getting into the cytoplasm. These trans-acting factors are quite abundant in the cell. In this slide, we're seeing here that there are about 2,000 genes which encode transcriptional regulatory proteins. So each gene, each regulatory protein can act on many genes, and then many genes can be regulated by one. Many proteins can regulate the transcription of one gene. So one gene could be under the control of many regulators, and each regulator can affect more than one gene. So this allows for fine-tuning of gene expression. And so it allows for gene expression to be regulated both spatially and temporally. So spatially, as meaning the cell type, for example, there's specific genes that are turned on in certain cell types and are turned off in others. And I gave an example on Monday, for example, in cells in the pancreas, cells located in what are known the islets of Langerhans, which are involved in producing insulin. That those genes, the insulin gene is turned on in those cells, but that same insulin gene will be turned off in, for example, cells found in the skin, because there's no need for your skin to be producing insulin. So multicellular organisms require that level of fine-tuning of gene expression. Here's an example of a cell-specific type of regulation of here we're looking at an enhancer that controls a gene that is involved in this case. In this case, it's involved in hair follicle or the growth of hair. Okay, and specifically the growth of facial hair in males. And we know that the growth of facial hair in males is under control of testosterone. That's a hormone, testosterone. And in order for this this type of regulation needs to be cell specific, because you don't want facial hair growth or hair growth occurring all over the body. So it has to be specific to that cell type. So here's an example of that at work. We have here an enhancer to which a transcription factor, in this case, this transcription factor is a receptor, binds to the enhancer only when the shape of it allows for correct binding. So the shape of this receptor changes in the presence of a steroid hormone, for example, testosterone. So the shape changes in the presence of the steroid hormone, and that change in shape allows it to bind to the enhancer. In the absence of that hormone, or the absence of testosterone, that receptor is unable to bind enhancer, and it will prevent increased levels of the protein necessary for making facial hair. This is an example of an allosteric interaction. That is where the shape of the DNA binding protein changes depending on the contents really of the cell. Okay, so here we have, this is an example of an allosteric interaction where the steroid hormone receptor binds to the enhancer only when it is bound to a steroid hormone. So that's an example of a cell-specific type of regulation. So how do these enhancers work? So we know enhancers are involved in modulating gene expression depending on what is bound to the enhancer. It could be, if an activator is bound, then it increases it above basal level. If a repressor is bound, then gene expression is decreased. But how does an enhancer know which gene to regulate? Because on last lecture, I mentioned that enhancers can be found upstream or downstream of the gene, upstream or downstream of the promoter, and it can be located at very long distances away from the gene that it's involved in modulating. So how does an enhancer know which gene to regulate? The answer to that is the presence of sequences known as insulators. Insulators are located between an enhancer and a promoter, and they would block the access of they block access to the promoter. So in this figure, it's showing you here an enhancer. And it's showing you two proteins. One is called GFP, or green fluorescent protein. Another is RFP, red fluorescent protein. And this enhancer theoretically can affect expression of either of these genes. But the presence of an insulator between the enhancer and the RFP, which is the red fluorescent protein, prevents that RFP gene from being transcribed. So insulators insulators located between the enhancer and the promoter will block access of the necessary transcription factors, or block access, yes, of the transcription factors, necessary transcription factors to the promoter, and hence prevent expression from occurring. So how does it actually work? The insulators help to organize the DNA into loops. So there is a protein known as CTCF, which binds the insulators, and that allows the insulators to create these DNA loops. If a loop that contains an enhancer and a promoter would allow that enhancer to modulate the expression of that gene via the promoter. However, if an enhancer is not located in the same loop as a particular promoter, it will not affect the expression of that promoter. So organizing the DNA into loops allows for control of the enhancer. It allows the enhancer to control regulation of a particular gene, and it does so via the insulator, because the insulator helps to organize the DNA into loops, and only enhancers and promoters that are in the same loop will interact with each other. An enhancer located in a different loop from a particular promoter will not affect it. Okay, so let's see if there are any questions regarding that. Okay, let's move on. I don't think there are any questions. Let's move on. So we talked about epigenetics. Well, the video, I should say, we didn't talk about it. Video talked about epigenetics. And epigenetics really is defined as inheritable changes that are determined by factors outside of the genome. Epi refers to above the genome. So factors outside of the DNA sequence. So epi means above. So epigenomics refers to above, or epigenetics refers to above the genome. So factors outside of the genome, DNA sequence specifically, outside of the DNA sequence, that can affect gene expression. So factors outside of the nucleotide sequence that can affect gene expression. And the text focused primarily on methylation as the primary mechanism used by epigenetics, the primary mechanism of epigenetics. So let's look at that. Let's see. Okay, so transcription initiation can be regulated by methylation. And DNA methylation is where you have a methyl group, and that's highlighted here in red, CH3. Our biochemists in the class should know that. So CH3 is added to a cytosine in a CpG, what is known as a CpG dinucleotide. So a CpG dinucleotide is anywhere you will find cytosine and guanine bound to each other. Okay, cytosine and bound to each other, not across from each other, not on adjacent, not on double strands. But when I said when the text refers to CpG, it's referring to cytosine and guanine that are next to each other. So whenever you have these cytosine and guanine dinucleotides occurring in the genome, the cytosine is methylated, or eventually will become methylated, as a way of controlling gene expression. So methyl group, DNA methylation, when DNA methylation occurs as a way of controlling gene expression, it's the cytosine in that CpG dinucleotide that is methylated. P refers to the phosphate. So you have cytosine, P, here's the phosphate, and guanine. So that's why it's written CpG. And it's written, the orientation is 5' CpG 3'. So that is the orientation that indicates, that allows, I should say, for methylation of the cytosine. The methylation is catalyzed by an enzyme known as DNMT, which is known as DNA methyltransferases. So DNA methyltransferase methylates the cytosine of CpG dinucleotides. In genes that are active, when genes are highly active, you will find CpG islands that are unmethylated. So CpG islands are regions where you have a high concentration of CpG dinucleotides. And in genes that are transcriptionally active, when genes that are turned on, genes that are active, these CpG islands are unmethylated. And they're unmethylated because the activators are able to bind and block access of the DNA methylases. And that allows for the chromatin structure to be opened. And we know that an open chromatin structure allows for gene expression. Okay, so methylation decreases gene expression. Gene expression and methylation are inversely related. So the more methylation you have, the less expression you have. And we see why. So when genes are unmethylated, it's because activators will block the enzyme DNMTs, DNA methyltransferases, from adding these methyl groups. And that will cause an open configuration of the chromatin, and hence it will allow for the genes to be activated, to be transcriptionally active. On the other hand, methylation of CpG islands will silence gene expression. So in the absence of activators, the CpG islands become methylated. They become methylated and that methylation causes the closed configuration of the chromatin. So there are proteins known as methyl CpG binding proteins that will bind to these CpG islands and hence prevent activators from binding, cause this close configuration of the chromatin, and hence decrease expression. So take home here is to know that there's an inverse relationship between, excuse me, there's an inverse relationship between methylation and gene expression. This methylation of these CpG dinucleotides is copied during replication, during DNA replication. So that although we're seeing here, there's no, we learned before that DNA, the information in DNA is replicated and passed on from parent to progeny. The methylation of DNA, that information, that methylation is also passed on from parent to progeny. And in this slide, it says here that DNA methylation is an epigenetic phenomenon because it can be inherited. It is heritable. It alters DNA gene expression without changing the DNA sequence. Any changes that occur that are not involving the DNA sequence are considered epigenetic changes. And so here it's showing you how that methylation is passed on from parent as the cell divides. See, we have a double-stranded molecule here, and both strands are methylated at the CpG dinucleotides. During replication, we know that replication is semi-conservative. We have our two new daughter molecules, each having one parental strand and one new daughter strand. The DNA methylase will then detect that methylation on the original parent strand and methylate the new daughter strand. So this methylation pattern is passed on from parent to progeny. Okay? There's a sex-specific methylation is responsible for a phenomenon known as genomic imprinting. Genomic imprinting is when the expression of an allele is dependent on the parent that transmits that allele. This is very different from what we learned all semester. We learned in the first chapter that the parent transmitting an allele or gene does not affect how that gene, it does not affect the expression of that gene in the offspring. If you remember, when we looked at the cross between purple and white flowers, the outcome was always purple, right? The dominant allele was purple. So a cross between purple and white flowers, F1s were always purple, regardless of whether the color purple was donated by the female part of the plant or the male part of the plant. We saw that the F1s were always purple. But in genomic imprinting, that's different. In genomic imprinting, it says that expression of a particular gene is dependent on the parent that is transmitting it. And this is under, genomic imprinting is under the control of methylation. Okay. Methylation occurs at regions known as imprinting control regions, which will silence gene expression. Because remember we said there's an inverse relationship between methylation and gene expression. And genes that are imprinted have regions known as ICRs, or imprinting control regions. And these imprinting control regions can be methylated. And if they're methylated, then it silences expression of that gene. Okay, so methylation of these imprinting control regions silences the expression of that gene. How many genes are imprinted in humans? Only about 100 genes are imprinted in humans. Most of our genes are not imprinted, and about 50 of them are paternally imprinted, and the other 50 are maternally imprinted. So paternally imprinted genes means that the paternal allele of that gene is methylated and hence silenced. Okay, and the maternally imprinted, a maternally imprinted gene, the maternal allele is methylated and hence silenced. Let's look at a pedigree, two pedigrees that demonstrate paternal and maternal imprinting. Okay, so the first part here, first part A, is showing you a paternally imprinting, demonstrates paternal imprinting. So here we see, remember, imprinting, you can equate imprinting to silencing. Okay, so imprinting means the same as silencing. So here we have a paternally imprinted gene. And so we have here a male, okay, and we see that only when, if a male has the mutation, it is not passed on because it's a paternally imprinted gene. Only when the female has it, that mutation, is it passed on to the offspring. Okay, so in paternally imprinted genes, the expression of that disease is not manifested when it is passed on by the male parent. You see, the male, although they have it in part A, a male who has the disease, he would only he will pass on the allele, but it is not expressed in his offspring. His offspring will be carriers of that gene, but it will not be expressed in them. So this is an example of paternal imprinting. Maternal imprinting is the opposite of that. So in maternally imprinted genes, the maternal copy, or when the mother passes it on to the offspring, it will not be expressed. It is silenced. So here we have a mother here, female, having the disease, but when she does not pass it on to her offspring. Here we have a male carrier, on the other hand, passing it on to his offspring. Okay. Again, male passing it on to the offspring, females not passing it on to the offspring. So that's a maternal imprinting. Okay, I will pause here and ask for any questions on genomic imprinting. Do I need to explain the pedigree any further? Okay, please explain further. Kimberly, let me go to Facebook. Okay. So, okay, let me try again. So imprinting is the same as silencing. When a gene is imprinted, it is silenced. If a gene is paternally imprinted, it means that if it's inherited from the male, if it's paternally imprinted, if it is inherited from the father, it will not be expressed in the offspring. When a gene is maternally imprinted, if it is inherited from the mother, it will not be expressed because imprinting is the same as silencing. Imprinting means silenced. So a gene that is, so looking at the pedigree again, in part A demonstrates paternal imprinting. So we have here a male. Here we have a male who has the disease. His offspring will not have the disease because it's a paternally imprinted gene. It means that men passing it on will not have, their offspring will not manifest that trait because it's paternally imprinted. A maternally imprinted is the opposite. A woman here, mother, although she has the disease or the mutation, it will not be expressed in her offspring. So imprinting, again, let's go back to the definition. Imprinting, it's a phenomenon in which the expression of an allele depends on the parent that is transmitting it, not what is received. It is only dependent on who is doing the transmission. Okay. Let me know if that was a little clearer. Okay, so I'm not hearing from anyone else. So I was assuming it's okay. So it's a little clearer. Okay, let's move on then. Okay, great. So genomic imprints are reset during meiosis. Genomic imprints are reset during meiosis. It says here that genetic imprints remain throughout the lifespan of mammals. The germ cells, however, the epigenetic imprints have to be erased and then reset during meiosis. So looking at this figure, here we have, because remember meiosis is the cell division that takes place that produces, I should say, your gametes. So here we have an egg and a sperm, and we know that egg and sperm are haploid. So we have an egg and a sperm, they unite fertilization to create a zygote. And you can see here the imprints on the egg, that's red, and on the sperm, there's an imprint there that's black. And here we have a zygote that contains both chromosomes, right? In the female, we know that somatic cell, there are certain cells that are designed to become germline cells. And here we have a germline cell that is diploid at this stage, but then it undergoes meiosis. And during meiosis, we have those imprints being erased. So here the imprint is erased and then reinstated. New imprints are made. So you end up with an egg that has the imprint that was started, that was in the original female or the original egg. Okay, so here we have that imprint is reinstated, and the same happens with the sperm. If this did not happen, then imprinting will not be sex specific. So this erasing of the imprints is necessary because here we have a germline cell that has both imprints. We don't want that occurring. Okay, so in order to prevent that from happening, you have to have erasing of the imprints and then reinstating of the imprints on the appropriate chromosome in the appropriate cell type, whether it's an egg or a sperm. Okay. So how does imprinting occur? There are two mechanisms by which imprinting occur. One is via the insulator. One is known as the insulator mechanism that helps to control imprinting. So the example we're seeing here is with a gene known as IGF-2. IGF stands for the insulin growth factor gene. So IGF-2, and it's showing you how the insulator plays a role in imprinting. So here we have the insulator. So the insulator we know is found between the enhancer and the promoter. Okay, so here's the promoter, enhancer, and insulator found between the two. If the insulator remains non-methylated, so that's what we're seeing here in the maternal copy, in the maternal strand or maternal inherited allele. So in the maternal inherited allele, we see that the insulator is unmethylated. An unmethylated insulator is functional, because remember, methylation decreases function, whether it's or inhibits function, inhibits expression, whether it's expression of the gene or it's expression of the insulator or the function of the insulator itself. So a non-methylated insulator is functional, and if an insulator is functional, it will silence expression of the gene. So here we have a maternal copy that is silenced or imprinted because the insulator is functioning. Okay. The reverse here in the paternal copy, the paternal insulator here is non-functional because it's methylated. So if the paternal copy is the paternal insulator is non-functional, you will have expression of the IGF-2. Okay. So again, a functional insulator would silence transcription of IGF-2, hence making this maternal copy in this example imprinted. Okay, so a functional insulator imprints the gene with which it's associated with, while a non-functional insulator became non-functional because of the methylation. A non-functional insulator will allow for expression of the gene, in this case, IGF-2. Okay. So that's one mechanism by which imprinting can occur. That's the insulator mechanism. And the other mechanism by which it can occur, imprinting occurs is via what is known as an ncRNA. ncRNA stands for non-coding RNA. So this non-coding RNA is found in the promoter of the gene. So the IGF, no, I'm sorry, let me take that back. This non-coding, the non-coding RNA known as AIR, A-I-R, is encoded, it is transcribed from the promoter found within the intron of the IGFR2 gene. IGF stands for insulin growth factor receptor. Okay, so we have an insulin growth factor receptor. This is just an example of the mechanism, imprinting mechanism. So we have IGFR2 intron, and in that intron, you have a promoter for a gene known as AIR, A-I-R. AIR is antisense because it's transcribed in the opposite direction of the IGFR2. AIR is antisense RNA, and that antisense RNA we learned previously will block transcription of genes. So this AIR antisense RNA blocks the transcription of the IGFR2 gene and hence silences it. Okay, so it says non-methylated paternal CpG islands allows for production of this AIR antisense, and AIR antisense will cause silencing of IGFR2. Methylated, however, methylated maternal CpG islands would prevent AIR from forming, that's the AIR transcript from forming, and would allow transcription to be active and allow transcription to occur. Okay, so those are the two mechanisms. We have the insulator mechanism for imprinting, and then we have the ncRNA mechanism for imprinting. Okay, any questions? Okay, let's move on. Here's an example of genomic imprinting in human diseases. So these are two examples, and it's actually quite interesting. So these two syndromes, one is known as Prader-Willi syndrome, and the other is called Angelman syndrome. And both of these lead to mental retardation and a whole host of developmental disorders. So what's the mechanism behind these two? This is, again, an example of genomic imprinting in humans. So on chromosome 15, on the Q arm of chromosome 15, so remember the Q arm is a short arm of the chromosome. So on the Q arm of chromosome 15, there's a region that involves several genes, but there are two, roughly two are affected by deletions in that region, okay? When the deletion is inherited from the father, it results in Prader-Willi syndrome. So in Prader-Willi syndrome, the deletion is inherited from the father, and the imprinted or the inactivated copy is inherited from the mother. So the father passes on a deleted copy of this region of chromosome 15. Again, so the father passes it on, but the mother's copy that is passed on is imprinted. Okay, so Prader-Willi syndrome would be an example of a maternally imprinted disease. Angelman, on the other hand, is an example of a paternally imprinted, because the deleted copy is inherited from the mother. The deleted copy is inherited from the mother, but the inactive copy, the imprinted copy, is inherited from the father. Okay, so this would be an example of Angelman would be an example of a paternally imprinted gene. And like it says here, both these individuals have mental retardation and other problems. You could read that in the text, the symptoms that are associated with this disorder. Okay. Okay, so these mechanisms are all examples of regulation occurring during transcription initiation. Okay, so imprinting affects transcription at the level of initiation. The binding of the transcription factors, the basal factors, the activators, repressors, those are all mechanisms that affect transcription or can regulate gene expression at the level of transcription. Now let's look at regulation after transcription, regulation that can occur after transcription. One of them being splicing. You know that after an RNA is synthesized, mRNA is synthesized, it is spliced. So the splicing and the stability of the RNA, the localization of the RNA affects the expression of that gene. So here's an example of how splicing varies and how it affects gene expression. And in this case, it's a sex-specific type of expression that is well, a sex-specific type of regulation, I should say, that occurs. So there's a protein known as sex-lethal, or SXL, and this protein found in Drosophila is required for female-specific development. Okay, required for female-specific development, and in the early embryos of females, so we know, step back, we know that females contain 2 X chromosomes, a male contain an X and a Y, and it's the presence of that extra X chromosome that allows for certain transcription factors to be produced. So in females, female Drosophila, they produce a host of activators known, transcription factors, 4 transcription factors, I should say, not a host, but 4 transcription factors that affect the production of a protein known as sex-lethal. Okay, without the presence of those transcription factors, no sex-lethal expression will occur. So again, in Drosophila, in both male and female embryos, you have this gene, the SXL. You have the SXL gene, but it's the presence of the X chromosome, having a higher number of X chromosomes, allows for certain transcription factors to be made. And these 4 transcription factors determine the expression of the SXL protein. Without those transcription factors, that protein will not be made. So we have here in the females, you have the protein being produced, but in males, that protein is lacking. Why is that important? Well, let me get to it. Oh, I think I missed that slide. Yep, I missed the slide. Let's see. 1 second. Yeah, here is the slide I needed to have there. Okay, so here we have this SXL protein being produced only in females, and what this XXL protein allows for is for more SXL protein to be produced. The presence of the SXL protein early in the development of female embryos allow for more of that protein to be produced. In males who are lacking that particular protein, they don't have enough or no additional amount of this protein could be produced. And this helps it affects certain characteristics and helps to determine whether or not an embryo develops into male or female. Okay, so that's all I want to mention there. We are running out of time, but I want you to pay attention to this slide on microRNAs and how microRNAs are involved in regulating gene expression, both at the level of post-transcription and also at the level of translation. Okay, so pay attention to microRNAs and their role in regulating gene expression, because I don't have enough time to go into that, but I think it's not difficult material to read. Okay, so any questions? Okay, no questions then. That's it, guys. Great having you in class. And if you have any questions as you study, please feel free to email me, okay? All right, that's it.